Hi, I'm Devatray, and I recently had the honor of interviewing Dr. Fred Jordan, the co CEO of a Swiss startup called FinalSpark. Their work in biocomputing is amazing because they're building these biological computers using human neurons and glial cells. A little recap after the interview.
Thank you so much, firstly, for taking out the time for this interview. It's really great having you here. Um, and I hope you're doing well on this fine day.
Yes, thank you.
Right. So, Final Spark, the name itself is electrifying, and your startup has been a lot in the news lately, and for good reason. You have been an integral part of the cohort of research in the field of biocomputing. So, could you please help us understand a little bit about your work, how exactly your human brain organoids work, and how you develop?
So yes, biocomputing is really about making a processor using living neurons. Okay. So normally in a processor you have transistors, and in our case we have human neurons. So that means first is biology. Okay. That means we have a cell culture lab, which is just below here, where we culture and multiply all those neurons, and then we connect them together, and we create a small ball, if you want, of nervous tissue, and then we put electrode on it, and then we communicate with it. That's all.
Interesting. So, organoid culture is the basis of your biocomputers, and they're really gaining traction in the field of biology and research in general because of the context that they offer, not just for biocomputing, but also drug delivery systems. So, what inspired you to work specifically on human brain organoids, and what got you interested in the field in the first place?
Okay, so now you talk about organoid. So organoid, indeed, that's the right word. These are organoid means like it's not a real organ, not a real part of the brain. Okay, it's somehow similar. Okay, and smaller, but so we don't say it's an organ. We say it's an organoid. Okay, like a bizarroid. It's not the exact real thing. So what led us to use living neurons for computation purposes. Okay. Um, so just the background is that I love science fiction. Okay.
Explains it.
But if you love science fiction, you're very naturally open to new ideas. Actually, you're seeking new ideas. Okay. You're living in the future. You have always a quest for new incredible things. So it's a natural tendency, but it's shared by all the fans of science fiction. Okay, nothing that original. Okay, and the other thing is that just by luck, we had one of our employee. So initially, we were doing only simulations of neurons. Okay, that means on a computer. Okay, we make simulations. By the way, this is the way ChatGPT is working, by simulating neurons. Okay. Well, basically, most of the AI today works this way. Okay. So we were working in this field, and one of our employee, just by pure luck, happened to work a little bit in the lab where they were measuring human neurons. Okay. So the goal of the project was really to characterize each type of human neurons, because there are about 400 different types of neurons in our brain. So, and at the same time, we had problems of powering our server for computation because our simulations were taking so much energy, like kilowatts of power. So we realized that the work that he was doing on living neurons meant also that living neurons is consuming way less energy than a simulation of a neuron. And when I say less, it's really less, like 1 million times less. Okay, and 1 million is a big number. Okay, we live in a world when you get a 20% gain of efficiency, you say, wow, that's wonderful. Okay. So here I'm not talking about 20%. I'm going to be talking about 1 million times more efficient. So it was like we have a problem as an engineer. We are consuming too much power for simulating our neurons, and we have a solution. The solution, the thing is, has not been done or built by human beings. Okay, it's our neurons. It's the result of hundreds of millions of years of evolution. Okay, so somehow when you use neurons as an engineer, you're cheating a little bit. Okay, instead of doing the things by yourself, you take something which is a result of very fine optimization. Say, oh, okay, that's actually very good. Let's use it. Okay, instead of trying to imitate it. So that was just the reasoning, very basic, very simple.
That makes a lot of sense. And there's this beautiful quote which goes, nothing in biology makes sense except in the light of evolution. And like you said, our neurons have evolved over millennia, more than a millennia, and they've really been fine-tuned for efficiency. So these silicon-based conventional computers and AI models, as you mentioned, consume a lot of energy, and they're not efficient in the long run, they're not sustainable. But your biocomputers are clearly a million times more energy efficient and a lot better at processing different kinds of information. So what would you say are the greatest strengths of the system that FinalSpark has developed?
Well, the greatest claims for FinalSpark, the thing is that this is still a real topic of research. Okay, so imagine quantum computers 15 years ago. Okay? And it's still a topic of research today, okay? But we are at the beginning, okay? So right now, the situation of FinalSpark is more being like a research tool. We work with researchers who are actually using our brain organoid remotely, okay? Not to do computation. We are not there yet, okay? But to do some research. So the nice thing is first, there are about 10 universities in the world who remotely 24/7 connect to our brain organoid and make some tests. So it's really open innovation. Okay, this is the stage where we are. And there is a new thing that has happened recently is, ChatGPT and large language model. Okay, they are very efficient when you discuss with them because they are based on the amount of information. But there is another thing which has happened recently, are AI agents. And AI agents, some of them are now starting to be used for research. So what we started to do is not only to have universities trying to interact with our neurons, but to have AI agents to which we give the control to interact with neurons, trying to change the way they are connected. And so they send autonomously some signals to the neurons. They measure the response of the neurons. They explain to us what they did, why they did that, what they concluded of the experiment, and then they decide for the next experiment. And they do a new experiment every 10 minutes. And we can run this 24/7. This is totally different from the traditional LLM, because in LLM, normally your base of knowledge is fixed. But this is totally different because here it's a closed loop with the real world, experimental closed loop. So it's a new kind of researcher who is working also with our brain organoid. And the goal here is really to train them to do something. Because you have to remember, when you create a brain organoid, you are connecting neurons randomly. Okay. So if you connect neurons randomly and you have a stimulation to them, they are going to reply randomly. Okay? And this is not useful. A useful system, for instance, you show a picture of a cat or a dog, and there are some neurons that will tell you if it's a cat or a dog. This is useful, input, output, okay? So the only way to solve this is to correctly tune the connections between the neurons. It's exactly the same for artificial intelligence. No difference, okay? We start with random connections and we tune them with learning algorithms. So in our case, we also start with random connections, but the learning algorithms are still unknown. And this is the main topic of the research of the universities and now of AI agents, is to find a way to stimulate the neurons in order that they learn something the same way as you are learning something right now. Okay, I'm giving an input to you, and it's possible for you by your output to show me that you understood what I said. So it's the kind of things that we do when we learn us human.
Right. That sounds fascinating. And what would you say are the biggest strengths and limitations of your system?
So right now, the most obvious limitations are lifespan. Okay, when we put the brain organoid on the electrodes that are going to live for a few months, or a few weeks, or a few days. So, and this is quite unpredictable. The number of electrodes also is limited right now. Okay. The types of neurons. So you have to imagine that we create organoids for different parts of your brain. So you can create brain organoids of your hippocampus, for instance, or striatum or cortex. Okay. So we are still limited in the type of organoid that we can create. And what we do actually are not only organoids, now are assembloids. So you take a striatum and a cortex organoid, you connect them, and you create an assembloid. So, but we are still some very much limitation. There are fundamental limitation in biology and cell culture. This is not new. This has been around for half a century. Okay, it's variability of your result. Okay, when we create two brain organoids, even if we take very much care of what we are doing, they are going to be different. So therefore, we have also to have algorithm which are able to scan with the electrodes which type of organoid that we have and how we should adapt to it. So there is a lot of things where we try to fight against variability, and on the other side, we try to find algorithms which are flexible enough to adapt. And the size, we are limited to very small, like a few millimeter of diameter right now, and we would like to scale this up. And well, if I can start to list all the challenges, there are a lot of them, but these are the big ones.
Right, I see. And last year in a podcast interview, you also mentioned that unlike your biocomputers won't be replacing traditional computers, but instead they'll have a very niche application, sort of like quantum computers do. Could you elaborate a little on that as well?
Well, I, I would not say that. Indeed, you're right. They're not going to replace computers. I wouldn't say that they're going to be that niche. Okay. They may actually replace silicon-based computers for running generative AI. Okay. It's a particular kind of thing. When you do ChatGPT, for instance, you interact as a human being at the speed of a human being. Okay. You don't need to have 1 million words per second. Okay. And brain organoid and biocomputer is going always to work at the same speed as us because it's they're made this way. So for all this that implies interaction with human being, real-time interaction, I think biocomputing could replace most of the silicon-based alternative today.
I see. Again, very interesting and intriguing. There's also another lovely quote by Francis Crick that goes there as follows, that the development of biology is going to destroy, to some extent, our traditional grounds for ethical belief. And it's not easy to see what to put in its place. So there certainly are ethical considerations when it comes to using human iPSC-derived neurons and glial cells. Would you like to address them, maybe?
Yeah, well, the word destroy is negative, okay? But I fully agree with the underlying idea that biology, biocomputing is going to lead us to rethink our view on living systems, all right? Because when I'm saying something as simple as I'm going to use living neurons, just I'm going to use a living neuron. It ends it, okay? So I consider this as a machine. And this is already disruptive. Actually, this is the only thing which is disruptive in this whole thing, okay? Um, Now, I'm just a technician, okay? And now you're talking to me about philosophy. Okay. I'm not sure that I'm very competent to answer to this, but the good news is that there are many people who are very competent to this. And so we started to connect with several universities in philosophy. Okay. And the goal is that they publish on biocomputing. So what we do is that we feed them with the scientific side, and they should make their thinking and think about this also on the ethical perspective, what people need to consider with this coming new industry. It's going to be a revolution, okay, comparable to a digital revolution. It's going to change the world.
Okay. Again, so you're connecting with a lot of people in philosophy to get the right perspective on this. And it makes sense because you, as a scientist, are treating, like you said, your neurons as simply a system, a machine. Right? Also, with all the time and effort and soul that you and your team have put into FinalSpark, I'm sure there were a lot of hurdles and challenges and lessons learned. So, if you could go back in time and you had to establish FinalSpark all over again, is there anything you would do differently?
Okay, that's an unusual question. um, I think we did pretty right because we had many people who helped us, and that was really cool. Remember, we didn't know anything about cell culture when we started. We directly talked with very renowned professors like Professor Potter, um, and I think we were really in the right configuration because we knew also a lab at the University of Geneva who was doing exactly what we need, called EPIA. Um, and they taught us how to culture cells, how to make brain organoids, how to measure them. Um, yeah, maybe on the type of electrodes, maybe we should have tested different types of electrodes. I'm not really sure of this. I really don't see mm, very big mistakes. I'm sure there are a lot of mistakes, and we are trying a lot of things, okay? And most of the time the things fail, okay? It's part of the research. But I cannot identify a big mistake that we would have done, but it's slow by essence, this kind of work.
Right. There are no mistakes, only happy accidents. And I'm glad, that there's nothing you would change. One final question. In the ever revolutionizing 21st century, a lot is changing in terms of research trends. And the students and young researchers in the field are increasingly interested in biocomputing. I myself am a student pursuing biotechnology. So, is there any advice you would give us students and young scientists?
So regarding at least FinalSpark, okay, we have an incoming stream of young people who want to work at FinalSpark. Okay, so first thing is that we have a Discord server where people can just post, and the team will answer to every question. Okay, to everyone. Now we don't have open position at this point at FinalSpark. However, we always have one intern working at FinalSpark, and we change intern each 6 months. Now, we also have candidates for all over the world for internship at Alvision. So what we decided after many hesitations, okay, was that we would take only um, students, interns from the NeuroX. I'm putting this in the chat. NeuroX from the EPFL, because this is a local school. When they really train students exactly to what we need, which is both neurobiology and AI. Okay, I mean AI, I mean programming. Okay, so you need these two things to interact with neurons. Okay, because we need to construct an interface, so there is a lot of signal processing and mathematics implied. Um, and now there are also IDs, people who can access to the neural platform. So we give free access to selected projects. Okay, so now we have 10. It doesn't matter the reference of the person. Okay, it can be a professor or it can be a teenage. I just don't care. Okay, what does matter is the quality of the IDs. Okay, and so for this we have a selection process and we always screen incoming projects. So this is another way to work in biocomputing, is to use our neural platform and try some things. So, and of course, this starts by reading the literature. There are not a lot of it, but we made a publication last year in Frontiers. So applicants should know about the science. So I think it's important.
Thank you. It's been an amazing interview, and I really enjoyed, speaking with you. And Final Spark is doing brilliant work, and I wish you all the best.
It's very kind of you.
Time for the recap. So, biocomputation is this booming new field that lies at the interface of computation and biology. And what FinalSpark is doing is that they are developing biocomputers using human neurons and glial cells. They're essentially taking any type of human cell, reprogramming or de-differentiating it into its stem cell version called the iPSC, and then redifferentiating it into any type of cell, in our case, the neurons and glial cells. They then perform something called organoid culture, which is this 3D culturing technique where you form balls or clumps, like Dr. Fred mentioned, of these diverse types of cell lines that you want to use. And these organoids are the closest thing we have to a working organ. They're not exactly like the brain, but they're close, you know, they're as close as we get. So they culture these in petri dishes and then they stick in electrodes for the transmission of electrical impulses, just like regular computers. The difference is that regular computers use a lot of power. And our neurons have evolved over a long evolutionary time scale to have perfected their efficiency and their power consumption. Our cells, including neurons and glial cells, rely on ion channels and essentially transport of ions and electrons to transmit these electrical impulses, making them a lot more efficient than the traditional computers that we use. And so these um, organoid cultures, they're not isolated. There's like a lot of them. And the whole system is called Neuro Platform, which, by the way, you can access through the website. And if you have an amazing project idea, you should definitely reach out because, like Dr. Fred mentioned, it doesn't matter whether you're a researcher or a professor or a teenage student, as long as you have a good idea, they'd be happy to, help you run their system. So one of the demos available also includes this butterfly that's initially randomly flying around this space of flowers, a patch of flowers. And this butterfly is essentially our organoid cells. And what you do as a user is that you provide electrical impulses through the web interface. And those impulses, they're provided to the culture through your electrodes. And you're trying to train these neurons and these glial cells, mostly the neurons, to respond to the electrical stimuli you're giving, essentially trying to make the butterfly um, go to a specific flower in a certain direction in a very directed manner instead of flying around randomly. And while this may seem trivial, it's actually a very big step towards how we work with AI and language models and all these intelligence networks that actually model regular neural networks and neural interactions. So the team really said, why should we, you know, go where the whole world is going and developing all these neuron inspired systems where we can literally work with actual living neurons? And that's exactly what they're doing. And the work is amazing. I'll put the links in the description. You should definitely go check it out. There's just so much we can do with these biocomputers, and their efficiency, like Dr. Fred mentioned, was a million times better. Essentially, they're consuming a million times less energy than your conventional computers, including the PC you're probably viewing this on or the phone. And that's going to be a very big breakthrough, considering how much energy, including fossil fuels, including water, and all the unsustainable after effects and the footprints that our current AI and language models are tarnishing the world with. So, again, brilliant work and more power to them. Go check out the links.